Welcome. I'd like to show you RipSeq Single. To access RipSeq Single, go to www.ripseq.com. Once here, you will need to log in with your username and your password. On the welcome screen, you will see on the right side product documentation, which includes the instructions for RipSeq Single. To access the software, click the RipSeq Single banner. This will bring you to the Single Files page. You can see a folder structure on the left side of the page. The current folder is selected. Files can be uploaded directly into this folder, or a new folder can be created by clicking the New Folder button. Add a name for this folder, and then click the Update and Save button. This folder will automatically be selected. Note that the primer set is already selected for this particular folder. To modify that, you can go to the Single Settings page and choose the appropriate primer from the drop-down list. Also on the Single Setting page is the interpretive criteria, which is used to create the identification. This is defaulted to CLSI guidelines, but can be modified to fit your laboratory. Next, we will upload the files. You can upload FASTA files or ABI files. In this example, I will upload a batch of ABI files using the Bulk Upload tab. From here, you simply navigate to the files that you would like to upload, select them, and click the Open button. The files are being uploaded into the system. They are being trimmed for quality, as well as the primers, and assembled into a consensus sequence. This will bring you back to the Single Files page, and you will see that the files have been uploaded with average quality scores, as well as the consensus length that was generated from both the forward and reverse sequence. The next step is to identify the samples. To do this, we will choose the appropriate database. In this case, we're going to use GenBank 1, which contains references with only valid species nomenclature, and then we'll click the Bulk Analyze All button. Results will appear in the table based on the interpretive criteria that is set in the Single Settings tab. Notice that the samples have an identification, percent identity to the top match, as well as an indicator flag. The green indicates a species level identification. On the top sample, a green with a slash call indicates that 2 or more species were identified for this isolate. We can look at the details by clicking the green arrow. The details page includes all the top matching references to our sample. Notice on this page is that there are checked references. These are the references used for the identification. Additionally, you will see links to the original GenBank reference, to up-to-date nomenclature at the DSMZ website, as well as any publications associated with this reference. In this section, we have additional annotations, which include approvals, rejections, any notes applied to a reference, references identified as type strains, or that are part of the Living Tree project. You can select any one of these references, and it will bring up the alignment under the table, as well as any notes associated with that reference. You can manually adjust the identification. You can do this by deselecting or selecting more references. In this case, we'll deselect this reference and click I verify this result. Notice that the identification changes for this isolate. The updated result will be saved. We can now go back to the single sequence page, and we'll notice that the identification has changed for this isolate, as well as there's a check mark indicating it's been verified. We can also verify samples directly from this page by selecting them and clicking the Accept Selected as Verified button. And that's how quick and easy it is to identify samples with RipSeq Single. Thank you.